So I'm going to tell you a story about dirt. You never know where you're going to find the newest discoveries. And we have colleagues who are undaunted by difficulty who have come up with a molecule that is the first ADAR1 inhibitor ever to be made that was propelled into the clinic by somebody who's in the front row. And this is named after her, Rebecca Morse. Thank you very much. Rebexinib is the name of the drug. And this is what happens when people never, ever, ever give up. Ganbate. And that is a Japanese term for never giving up, right? So we're all here to talk about how we get stem cells that go from aging to precancer to cancer stem cells, and how do we unravel this complex puzzle and actually intercept it before people go from a precancerous condition like a myeloproliferative neoplasm that you heard Noah talk about to a full-blown cancer, and how much of this is human-specific biology that's actually stress-induced. So when I'm in clinic, people say, how much did stress have to do with me getting this disorder? And so we really want to unravel this. Unfortunately, we can do this in the hematopoietic or blood-forming stem cells because they're readily accessible. As you heard Allison say, it's hard to get a piece of your brain. People don't really want to voluntarily give that up, but they will give up some blood, in some cases, some bone marrow. So we've been working on how these disorders where you make too many of one type of blood cell, whether it's white blood cells, red blood cells, or platelets, first start. And they often start at the level of a hematopoietic stem cell or blood-forming stem cell. These were discovered in 1961 by Tillam McCullough in Canada because people were worried about nuclear proliferation. And they thought that radiation would wipe out our entire bone marrow. Then Tillam McCullough discovered these cells called hematopoietic stem cells, and Irv Weissman was able to find the surface markers that allowed us to isolate that. And since then, that was back actually in 1992, we've been uncovering where these mutations start. Why do we care about stem cells? Stem cells are supposed to regenerate our tissues, but they can also regenerate cancer if they've gone awry. So we've been targeting the pathways that allow these stem cells to take the initial step between being abnormal and actually being able to clone themselves. So I'll talk specifically about inflammation-dependent base editing enzymes and their role in myeloproliferative neoplasm progression to acute myeloid leukemia. This all starts with these stress enzymes that are human-specific. So humans evolved to protect our stem cells from repetitive elements, these retroviruses like human endogenous retroviruses and long interspersed nuclear elements, lines and HERVs. Now, this is biology that was ignored for a long time because we couldn't sequence it. They're repetitive elements. They were really hard to see, but they're very, very important for telling our stem cells how to behave when you're undergoing stress. So they tell us to mobilize our stem cells and they tell our stem cells to clone themselves. But when they do this too much, the enzymes that were supposed to prevent this active retrotransposition or activation of lines and HERVs actually start to mutate our DNA. Those are apobecs, or our RNA, that's ADAR. So we have these repressors of retrotransposons or repetitive elements, that dark genome, that when they're activated for too long can actually lead to precancer and cancer. So why does this have anything to do with space? So like Allison, I was looking at the NASA twin study. This was published in Science in 2019. You see that Scott Kelly came back 2 inches taller than his brother Mark. Amazing. You get to be taller. Apparently your skin gets better. According to Peggy Whitson, you lose your wrinkles. The only thing is, Scott Kelly, after almost a year in space compared to his brother Mark that didn't spend much time in space, came back with immune dysfunction, pre-malignant changes in the blood that looked like they were just the beginning of a pre-leukemic disorder, and the same inflammatory growth factors that we know can activate these enzymes, ADAR and ApoBec, that can mutate our RNA and DNA respectively. So now we've completed 9 missions to the ISS. We're thrilled to be able to work with our implementation partners and NASA to be able to see in an accelerated time frame how stem cells age, how they become pre-malignant and malignant, and that's something that is all detectable now thanks to the work we've done with NASA and our implementation partners. So we've developed these canaries, as I like to call them, the Cancer and Aging Stem Cell Nanobioreactor, Artificial Regenerative Intelligence Enhanced Systems. My dad was a coal mining engineer, and he always taught us that you take a canary into a coal mine because they're much more sensitive to carbon monoxide. And so this is our equivalent. It's like a canary or a carbon monoxide sensor of stem cell stress. We made this with Space Tango. So we've got the Space Tango here team today. Hey, Twyman, Paul, we've got a number of you here because we just did de-integration of SpaceX 33 for us and Alison Motri's mission as well. And so basically, this is a little incubator. It's a cube lab. You see here, you've got this flask for the media. You've got the nanobioreactors that we made here. All of these experiments have been directed by our stellar mission specialist, Jessica Pham. And basically, we can put stem cells in these little nanobioreactors and then image them with this microscope. So we can see when stem cells are asleep. They're supposed to be asleep 80% of the time to maintain their full activity. When they're in cycle, they're green. When they're dividing, they're yellow. You see how small this pediatric blood bag is? That has all the stem cells we need to actually reproduce all the blood cells in our body if we had to use this as a therapeutic avatar, or theratar, as I'm calling it. This is a splicing reporter, so we can see in real time in space and on the ground whether or not the stem cells are splicing too much. They turn green. And then we can see, have they activated this malignant gene called 8R1, which is here in green, in tumor organoids. So we did this all so we could see, are we seeing accelerated stem cell aging in space? As was suggested by Scott Kelly, coming back, looking about 10 years older than his brother, at least from a blood standpoint. So we did this study, NASA funded, with Space Tango. We were able to see that stem cells age faster, probably 10 years faster in space than on the ground. And the most notable finding was that in flight, there was activation of what we called the dark genome, with the cells that had been in flight here in red compared to ground controls. So we talked about activation of the dark genome, or Darth Vader of the genome, because you inherit it. And that seemed to catch the attention of CNN, NBC, all these other groups, because they thought this was cool. The thing is, we didn't just invent this, we have this. Our DNA is littered with all of these vestigial viruses, retroviruses, and they have been part of our genome for thousands and thousands of years. They should stay quiet, but they get activated under conditions of stress, and they lead to this stress biology that can induce accelerated stem cell aging and precancer. So we were feeling very Debbie Downer about this whole thing. And we thought that once aging has started, there's nothing that can stop it. This is a freight train. But actually, this is thanks to our friends in the audience from Axiom. Pinar, hello. Here you are. And our biorepository team, Inga, again, Jessica, the whole lab. We were able to study whether this is true in astronauts. So we looked at the Axiom 2, 3, and 4 crew members. They're all superhuman. They all seem to have this idea that everything will work out. So I don't know if this is part of astronaut training or whether they were just genetically built to be like this. But we decided to ask the question, do they age faster from a stem cell level in space? So we looked at Axiom 2 crew members who had been there for 10 days, Axiom 3, 21 days, Axiom 4, 20 days. And so we were able to look at before, during, and after return from space to see what their stem cells did. So we're able to do this for all of us now, by the way. So if you wanted us to look at your stem cells in your blood to see if you're stressed or not, we can actually do it. We can take 2 little vials of blood. We can separate out the purified stem cells that are CD34 positive, and then do whole genome sequencing to look for mutations with Ludmila Alexandrov's lab, whole transcriptome or single cell RNA sequencing, and then look at aging of those stem cells. So when we first looked at the data, I thought, well, here we go. We're seeing accelerated aging. But then Patrick Chang, because I always love to point him out, and Claire Engstrom in the lab said, actually, there's this other population of stem cells that comes up in flight in response to stress that is a more primitive stem cell. And I remember saying to Patrick and Claire, that's not a stem cell. It looks too primitive. But it turns out, just a little while before we made this discovery, Dominique Bonnet found this population of EPCR positive stem cells that is 10 to 100 times more capable of regenerating our bone marrow. I just hadn't read the paper. So went back, read the papers, and holy moly, this is what the astronauts are doing. They're mobilizing this highly regenerative stem cell population in response to stress, not just space stress, but they even do it a little bit on the ground in anticipation of launch. So this is something that we can test in all of us now, and it's a marker of resilience. And it turns out that that stem cell population may outcompete the mutated stem cells. And those stem cells we think of being mutated by activation of that enzyme I mentioned, ApoBec3C. So we're able to see, are your stem cells revving up and regenerating, or are they being corrupted by this enzyme ApoBec3C and getting mutations that we can detect? 10 days in space is not so bad. 3 weeks, we start to see mutations. But for people that can mobilize this regenerative stem cell population, it seems to outcompete the bad. So how is it that we can detect and expand those cells and prevent precancer from becoming full-blown cancer? One way is to detect ApoBec3C and reverse that. We're working on it. But we decided to start at the end of this story, target this malignant enzyme that works in a human-specific way to corrupt our stem cells, and that's called ADAR P150. The reason that's so bad is it allows cancer stem cells to clone themselves and evade the immune system. Now, ADAR1 is something that our lab discovered back in 2012, actually, as something that allows cancers to clone themselves. This became something that a lot of groups worked on and started to look at ADAR high as bad in AML. If you're high in ADAR, you don't live as long with AML, multiple myeloma, liver cancer, breast cancer, 20 different cancers if you activate this. But it wasn't just a story about cancer cloning itself. It was a story about cancer evading the immune system, because if ADAR is turned on, it turns off interferon in the tumor microenvironment, so the T cells go, okay, let those guys go. It gives the cancer a hall pass. So we started saying there's no time to wait. We have to strike at cancer just as it's getting started, or ideally before it really takes off, blasts off, pun intended. So here's Jessica working at ISSPF, preparing our tumor organoids for flight. We started testing cancers in space with Axiom-1. We showed that the tumor organoids tripled in size in just 10 days. But perhaps most importantly, in addition to tripling in size, they activated that enzyme ADAR1. And that gave us an opportunity to target this enzyme. So here's Walter Villaday. He's an astronaut. He's doing our experiments with breast cancer organoids. He's comparing two drugs, fedratinib and rebexinib. And that rebexinib is the drug that very specifically prevents ADAR P150 from being activated, whereas fedratinib is a weak ADAR inhibitor. So we wanted to compare the two. And basically what we found in that study is rebexinib behaved as a cancer kill switch. If you're not growing, you're dying. The tumor organoids just stopped growing. So then we said, well, that was in a cell line. What about metastatic breast cancer from patients? And we found, this is day 4 of our Axiom-4 mission, the rebexinib stopped the growth of these tumor organoids. They have that ADAR green fluorescent protein reporter in there. Looked very similar to genetic knockdown of ADAR. But I think the surprise was really glioblastoma multiforme. So GBM is the most malignant brain tumor in adults. It affects 13,000 people annually. It has a 100% mortality rate, usually within 18 months. There's nothing that works to stop this freight train. So we were amazed when rebexinib at low doses seemed to stop this tumor from propagating. And, the ADAR knockdown wasn't even as dramatic. The other disease that responded exquisitely sensitively was secondary AML that has evolved from a myeloproliferative neoplasm. So you see this forms this almost solid tumor cluster of disease. This is day 5 in flight. It had just stopped growing and couldn't be propagated. And then Jessica and Wen showed that if you transplant the cells that have come back from flight and were treated with a vehicle control, they still grow the leukemia, but the ones that were treated with rebexinib have trouble doing that. So we've got an active investigational new drug application now. The company Aspra will be taking this forward to that phase 1 clinical trial with rebexinib for treatment of patients with myelofibrosis or secondary AML. But the idea is that it could expand to a number of other ADAR-activated malignancies. And the way to accelerate the implementation of this more expanded trial is because of space. We only got this accelerated view into how cancers evolve because of our NASA ISS missions. So basically what we're looking at are these twin crises in cancer. The number one cause of death from cancer is actually the cancer comes back. And the reason it can do that is our stem cells get exhausted. They don't make a proper immune system and they can't see the cancer anymore. So we're tackling these twin crises in cancer with our NASA crew members and the Undaunted Axiom crew members. We're thrilled to be able to work with Redwire, Space Tango, Axiom, Bioserve, and we just signed an MOU with VAST, so we're thrilled about that. Thank you, Ken, for propelling us forward together with Meg and the whole team at VAST. But the point, I guess, of all of this is to say what Noah was telling us. It's our collective responsibility to make sure that never should there be a person that has to worry about the cost of care. We need to make therapies more effectively. This drug was found in the dirt in Japan. It's a bacterial metabolite. So it's a soil bacterium that Jim LeClaire and Mike Burkhart were able to purify. But this is the dawn of a brave new era where we can do this bigger, better, faster, smarter. And faster includes spaceflight. So I just wanted to thank everyone for being here and for recognizing that this is just that pivot point in time where we all have to do this together. Thank you.
Amazing talk as always. I guess my question is on EPCR. As you know, I used to work on this, and you said that it's kind of resilience, that it's part of the resilience that may be kind of expressed on them. And so I guess my question would be, hey, what do you think the mechanism that would lead to that resilience? Is it just a stress? Is it something else that, the bone marrow of the science is sensing? And then the other way, we know maybe ways that we can mobilize those cells. Will that be actually a way for us to increase resilience, or would we want to avoid it actually?
Yeah, all what we're working on now. So it looks like there are 3 populations in there. There's one that doesn't express APOBEC, so it's not a pure population, so it doesn't look like it can mutate itself as readily. We do think it is sensitive to a mechanosensor called piezo-1, and that is something that Fubi in the lab has an agonist and an antagonist for. It's a way of expanding this population. It's also ADAR high and line and HERV high. So not all of these retro elements are bad. It's just when they're activated for too long. So they are part of normal regeneration. So we do want to learn how to expand them. You certainly have your whole Ph.D. on this. So yes, please, we'd love to work with you. But we don't think this is just for blood stem cell biology. We think it's going to be important very early in neural stem cell biology, skin stem cell biology. Cancer, takes a highway. It hijacks these stem cell pathways. So we're trying to dial it down where you see it in cancer. But it could also be the other side of the coin where we can enhance regeneration, provided we can control it.
For the drug regenerative, where does the bacteria? You said it comes from dirt in Japan. Okay, who found this bacteria in dirt in Japan? That's really interesting.
It's such a great question. This comes down to basic science. There was a scientist working in Japan that said, wait, what's this? This drug seems to kill cancer, and was working with cell lines. And then a Japanese company, ESAI, decided, well, maybe this will be something. But it turned out it's really hard to make. We had no idea. When you start on this difficult journey, you never know how hard it's going to be. But a friend of mine who actually invented fedratinib, that drug that's now known as Inrebic, said, Kat, when you have a problem, go back to the beginning. Kind of like when you lose your keys or lose your cell phone, where does it come from in the first place? So it turns out that bacterium has an enzyme that makes the drug. So we were able to find the enzyme, clone it, make it fully synthetically. So I think, as Allison was alluding to, we have a lot of natural products, and go back to them and say, can we make this even more effectively? So it was a very clever thing. It's kind of like immune checkpoint inhibitors. They were discovered 20 years before that Nobel Prize was given. So some of these things take a while to incubate. And the point with precision medicine now is we can see it faster and we can develop things faster. I think we just need to put our minds together and be open-minded about the next set of cancer curing drugs.
With these experiments in space, At what magnification or what fold do you expect drugs to get approved? How much faster?
It's such a great question. And I was on a full day workshop with the FDA and NASA. So the FDA wants to regulate this. They're seeing things are happening very quickly. And so, as you can see, the FDA is really trying to look at approvals based on organoid models, tumor organoid models that started April 10th of last year, where they said a lot of these immune-based therapies actually aren't predicted based on animal models, and we can predict them based on patient-derived little mini tumors. So they're in it to win it. And I think when they talk about a fast path or acceleration fast path to approval, it can be as little as 3 years from first patient treated. So I've seen that with one drug called Sprycel, where it was 3 years from first patient treated. And I know because I treated one of the first patients when I was at Stanford. But I think we can really demand that this be done faster. And I think the reason it's faster is patients are really thoughtful and are giving their blood, their bone marrow, their tumor tissue, sometimes sequentially. And that's allowing us to see how cancers evolve over time and actually dial back the clock to precancer and say, wait a minute, this started at this precancer stage. So now we have genomics and RNA-seq, and we can make it very precise and have a stellar stem cell fitness portal, the Stellarnome, that you'll hear from Peter DeVolt about from Epic. So I think we should all have access to these data in real time. And I think it will accelerate approvals pretty dramatically. So you think about, usually it takes 10 years minimum to get from an idea to an FDA-approved therapy. We're trying to shorten that timeline to 3, hopefully. And with AI, we'll be able to get some early indications of what's likely to work. But it's not going to replace natural intelligence, which is regenerative, neural stem cell based, and we network because we bounce ideas off each other. So thank you.